And everybody say hi to Amanda. Oh, did the kitty. Hi, kitty. How you feeling, Amanda?
Like I got ran over by a car.
Can you hear me?
Yeah.
Good. Okay.
Honestly, like the worst part too is the injection site is like I can barely move my arm. It's super swollen and red and like I have to keep icing it.
Okay. Well, just take it easy, and maybe I will use this as a makeshift blackboard here. All right. Yeah. Yeah, and there's a light that's on it, which means all I need to do now is get some paper. I'll be right back and then we'll get started. Thanks for leaving that camera in there. It's really helpful. All right, we are ready, I think. Okay, Amanda, can you see the slides?
Yeah.
And do they move?
Yep.
Okay. And do they move?
Yep.
Good. Okay. All right, we're going to start. Let's go ahead and do the quiz first. So I'll give Mrs. Schaefer a copy of the quiz. Just come in and take it Monday, okay?
Okay.
And the quiz is right here. Okay, we'll get started. We were talking about high density lipoprotein particles. So high density lipoprotein particles, again, are made by the liver and also by the enterocytes of the small intestine. They are a regulatory particle, and their goal, their role, is to mop up excessive cholesterol in serum and from the surfaces of cells, and also from that cat, and also from the plaques that are formed in vessels. It was thought for years that when plaques formed, they formed permanently. Now it's very clear that plaques can shrink. And it's very clear they do so by means of an HDL-dependent mechanism. So they take unesterified cholesterol, they use this LCAT enzyme, the lecithin cholesterol acetyltransferase. They use phosphatidylcholine to convert unesterified cholesterol into a cholesterol ester. And then they make the lysolecithin. Lysolecithin, incidentally, is a pro-survival cytokine for cells. Just a little tidbit of information. So it's clear that HDL particles can take cholesterol, excess cholesterol, from VLDL or LDL or even IDL, and they can convert it into cholesterol esters that the HDL then takes up. Two of the proteins that HDL is used to do this is ApoD and then this CETP protein, cholesteryl ester transfer protein. This is a protein that lets HDL steal cholesterol esters from circulating VLDL or LDL or IDL for that matter. So this is one of the reasons why large amounts of HDL is protective against atherosclerosis, because it tends to deplete circulating lipoprotein particles of cholesterol. Free serum cholesterol usually comes through an albumin-bound intermediate, and that will initiate the LCAT enzyme to convert free cholesterol into cholesterol ester, which it will store. And I didn't mention this, but as the HDL takes up cholesterol, it transforms. So it starts out discoidal, and as it takes up more cholesterol, it gets transformed into something that's spherical, like that, except prettier than that. Now, HDL can also take up cholesterol from cells and also from plaques and things like that, from foam cells, what have you, by means of a cholesterol efflux protein called ABCA1. ABCA1. So ABCA1 is a cholesterol efflux protein. And those hiccups. I'm going to go to our little makeshift blackboard here. So ABCA1 this particular protein will take cholesterol from inside cells and in exchange, well, actually with a hydrogen ion, it will pump out a hydrogen ion with the cholesterol. That's what this ABCA1 does. HDL very clearly can bind to ABCA1. So if we put another ABCA1 over here, and let's get an HDL particle over here. HDL has the ability to associate with ABCA1, and it can then use that cholesterol efflux to fill itself. So essentially it can relieve cholesterol-laden cells of excess cholesterol. Mutations in the ABCA1 gene that encodes this protein lead to a condition called Tungier syndrome. And Tungier syndrome is characterized by very high serum cholesterol levels and very low levels of serum HDL. And ABCA1
is ABCA1 short for anything? Is it like an acronym at all?
it's a member of the ABC cassette transport proteins. So this process is ATP dependent, but by effluxing cholesterol, it's very clear. This comes mainly from work done in rodents, but also from mutant studies in humans. In rodents, if you increase ABCA1 levels artificially, you increase the amount of HDL. You also increase the efficiency of HDL, and you greatly decrease serum cholesterol levels, and you decrease the tendency for atherosclerosis. In humans, there are different alleles of ABCA1, and those alleles, given how well they work, seems to correlate well with the amount of HDL and decreased risk of atherosclerosis. So human and animal work both tends to confirm this hypothesis. Tangier syndrome is due to loss of function mutations in ABCA1. You get low HDL, and you get increased risk of atherosclerosis. Can't see that very well, can you? Oh, it looks better there than it does here. Amanda, you look awful. You're pale. Your sound's off, but you just,
Oh, I said I'm running a fever. It's super fun.
yeah. You'll be fine.
I will be. They said it lasts about 24 hours.
Yeah, that's what it did for my wife. When my wife got Moderna, but that first day, she said, "Oh, I don't feel so great." I said, "Hon, just stay home." So she went to bed, and when I came home, she was still in bed. But the next day, she was fine.
Yeah.
Completely fine. And yeah, so, and you'll be immune, and you'll get to make fun of the rest of us who aren't, and yeah. All right. Any questions about ABCA1? All right, so ABCA1 helps expel the free cholesterol, and ABCA1 pretty clearly plays a role in clearing cholesterol from plaques. And that comes both from work that's been done in the test tube and also from work mainly in rodents. So HDL will help vacuum up free cholesterol from the surfaces of cells. And this other process seems to be ABCA1 independent. And what the cell seems to do is it seems to pump the cholesterol into the membrane. And if there's excess cholesterol in the membrane, the HDL will remove it in some sort of efflux fashion. And how it does this is not completely understood. So here we have essentially, this is from a review from Nature Reviews Immunology that looks at macrophages and the ability of macrophages to essentially take up and then get rid of excess cholesterol. So you can see there are all these receptors that can take up both modified and unmodified cholesterol. Then that cholesterol itself can also be effluxed by ABCA1 and then by this other protein, ABCG1, both of which can transfer the cholesterol to HDL particles. This seems to play a role in decreasing the tendency for the macrophage to become a foam cell. Whereas the uptake of cholesterol, of LDL, either by LOX1, CD36, the scavenger receptor A1, scavenger receptor B1, or by just penocytotic processes, these essentially make the cells become very cholesterol laden. If the cholesterol crystals out, you induce an inflammation. Cholesterol crystals, you ever seen cholesterol crystals? They are quite sharp. They can, yeah. Yeah, they can. They don't tend to crystallize in kidneys because they usually get bound up with other things. They usually crystallize once they come out, or they'll crystallize in the bladder. But then because the bladder is being washed through with liquid, now if you have some sort of condition where you're not able to void your bladder, then you'll get crystals, and those crystals can damage the bladder. And that's why, plus it increases your risk of infection, bladder infection. But that's why not being able to void your bladder is bad, very bad. Well, they don't look like that. They look more like caltrops. We're guys, we never played jacks when we were kids. The gals usually play jacks, at least when I was a kid. Did you ever play jacks? Well, actually, jacks is very good for hand-eye coordination. My daughter played jacks with her tennis team because that helped with hand-eye. And it looks like jacks, essentially. They're sharp. Yeah. Bladder cancer. You also see them in bladder cancer because of bladder cancer, the cells are sloughing a lot of junk into the urine and you get cholesterol crystals. And Dane, I don't know if you heard the question. Dane asked about cholesterol crystals in urine because you do see them sometimes. And they look like jacks, like from that game with the rubber ball and the jacks. You bounce the ball, you pick up jacks.
It just sounds painful.
Yeah. Okay. Now, what happens to the cholesterol that the HDL picks up? It gets taken to the liver and is converted into bile salts. So HDL essentially will traffic this unused cholesterol back to the liver. In the liver, it gets converted into bile salts. Then the bile salts go out to the GI tract. So this picture here is meant to illustrate this pathway whereby you go from peripheral tissues to ABCA1, HDL takes it up, HDL takes it to the liver converts it into bile salts and excretes it. Also, it can pick up cholesterol from VLDL and LDL. These, of course, can be taken up by the liver. That cholesterol that gets taken up by the liver will either be used for cholesterol to make new VLDL or it'll be converted into bile salts. So bile salts are the dumping ground for excess cholesterol. This process whereby you take excess cholesterol, return it to the liver, convert it to bile salts, that's called reverse cholesterol transport. Reverse cholesterol transport. Right, taking the excess cholesterol, taking it back to the liver, converting it into bile salts, that's reverse cholesterol transport. Now, one of the go back to the blackboard here. One of the big concerns in dietary science these days are these things called trans fats. So where do trans fats come from? Your textbook has a really good explanation about the origin of trans fats, which I highly recommend that you read. But Reader's Digest version, essentially. So you have unsaturated fatty acids that are in foods. Now, if this food is meant to be stored, this is subject to oxidation because the oxygen adds across the double bonds, and then you get aldehydes, and then short chain fatty acids and other organic molecules. These aldehydes are reactive, of course. They can react with other things in the food. And these aldehydes, plus their derivatives and these organic molecules, they make the food taste and smell rancid. So how do you increase the shelf life of the food? You reduce these double bonds. So how do you reduce double bonds? You've done reductions of double bonds in organic chemistry lab. You do a hydrogenation. And this hydrogenation, if you have a double bond, and you add hydrogen across it, well, of course, then you'll end up with a saturated fatty acid. Now, wouldn't it be nice if it worked that way all the time? Sometimes this reaction only occurs partially. And instead of reducing the double bond, you reduce it and then it goes back to being a double bond. But instead of being in the cis form, it's now in the trans form. Like that. And this is what we mean by so-called trans fat. Fatty acids essentially with these trans double bonds. Natural fatty acids that are unsaturated tend to be in the cis formation. Now, when you have food that has this sort of thing, your body kind of looks at that and says, I think we can deal with that, and it can. But when you have high quantities of these will tend to inhibit reverse cholesterol transport. Therefore, these can send your LDL levels into the roof. And that's one of the reasons why there's a movement now to try to remove trans fat from food. And so people now are being told not to buy so-called processed foods. That is, foods that have been adjusted to increase their half-life. So now you have foods with shorter half-lives. So there's a movement now towards fresher foods. And there's even another movement, which I really think is stupid, but the raw food movement. If you've read any of their stuff on the web, I see Amanda smiling. She's heard of that movement probably. And she's holding her nose. So the raw food movement takes it a step further and says cooking the food in some way, alters it in such a way that it's not good for your digestion, for nutrition and what have you. That is actually something that's been very heavily studied. That is in science what we call, what's the technical term? Crap. That's what we call that. Crap. So yeah, the raw food movement has basically nothing to it at all. No, they don't eat anything cooked.
There are some people who like steam broccoli and boil chicken. Even that's good.
What's wrong with steamed broccoli?
It's not that bad, but they're like, but you can't cook it.
I like steamed broccoli.
I like it sometimes, but sometimes.
Steamed cauliflower.
That's good.
That's good.
But I like
That's good. It keeps it doesn't have the bitter flavor of raw cauliflower, and it's got that little bit of toughness to it. And if you boil it, sometimes it falls apart. Steamed, it stays together. Terrific.
I like
Baked is good too. Baked is good too.
Only better to steam. Steamed is
Steamed, I like steamed.
Right.
Steamed is very good. Yeah, there's you see all kinds of things on the web. Yeah. I should show you a Twitter feed. I won't show it to you now. There was a lady made a statement to me. She said, there's no empirical evidence that masks work. So I said, no evidence. How about this experiment? No evidence. How about this experiment? Reference. No evidence. No, this is on Twitter. No evidence. How about this experiment? No evidence. How about this experiment? Then her response was, that's anecdotal evidence. See, when you're that, I'm done. You're not listening. I'm done. Talking to you is a waste of time. I'm done. So, kind of, sort of, yeah. So, okay, so HDL can also be taken up by macrophages. They can then take it up and then resecrete it. And if they do that, they can enrich it with ApoE. And that ApoE increases their uptake by the liver. This is called retroendocytosis. This has been shown in not just cultured macrophages, it's also been shown in both rabbits and in rats and mice to occur. It's been shown to occur in human cultured macrophages. I don't think anyone's shown it in vivo, but that'd be very hard to do. And then it can interact with several other tissues, not just the liver, because there's some tissues that need greater amounts of cholesterol, and they can actually get it from HDL. So for example, kidneys. Kidneys can actually take up HDL. They will metabolize smaller HDL particles. And kidneys, remember, also have adrenal glands, which use cholesterol to make their mineralocorticoids and their glucocorticoids. This is one of the ways they get enough cholesterol to be able to make those hormones. And it's not on this slide, but gonads also can do that as well, because they need cholesterol to be able to make testosterone, estrogen, progesterone, androsterone, things like that. And this receptor, this scavenge receptor B1, that's not BI, that's meant to be B Roman numeral 1. This will also cause the transfer of cholesterol esters from HDL to the tissue, which will deplete the HDL. This is one of the ways that some of these tissues can take up more cholesterol if they need it, because the scavenge receptor B1 is actually fairly widely distributed. And so this are just a few of the different types of organs that have this scavenger receptor. Of course, the liver has it, macrophages have it, platelets have it, all steroid making tissues have it. The adrenal gland is shown here, but also the gonads. And another tissue that actually uses cholesterol, but there's actually a lot less research on this, is the brain. The brain's use of cholesterol is extensive and it's different than other tissues. And it is poorly understood. There are some of these other receptors we've talked about are in the brain, but the brain has some other receptors for these particles. And then certain alleles of ApoE, ApoE4, for example, play a role in the onset of Alzheimer's. That is a link that's, again, not terribly well understood, but that is a wide open field of research that is just waiting for some good labs to go in and just really look at that. So that maybe, hopefully in the future, I'll have more slides about what goes on in the brain, because that's a little bit of a black box. when it comes to cholesterol metabolism. All right, so then we have the reverse cholesterol transport. We already talked about. Steroid hormones are made from cholesterol largely by side chain cleavage, side chain oxidation. That's a very interesting, albeit complicated process. We have talked already about the regulation of cholesterol synthesis. Remember, HMG-CoA reductase is the linchpin. It is the absolute linchpin for control of cholesterol. And insulin activates HMG-CoA reductase by activating its dephosphorylation. It's dephosphorylation. Glucagon activates the phosphorylation of HMG-CoA reductase through protein kinase A. That inactivates it. And in high levels of cholesterol, we saw that these high levels of cholesterol will tend to stimulate the ubiquitination of HMG-CoA reductase, which will then cause it to be trafficked to the proteasome and degraded. ACAT is also stimulated by high levels of intracellular cholesterol. That will make more cholesterol esters. More cholesterol esters will stimulate the formation of VLDL, because you have to package those into something. So if you have mutations in adaptins, so we saw one of those adaptins is called autosomal recessive hypercholesterolemia, ARH. Then mutations in the LDL receptor. There have been plenty of mutations mapped in the LDL receptor. These will lead to genetically determined high serum levels of cholesterol. These are called familial hypercholesterolemias. They can prevent synthesis of a functional LDL receptor. They can prevent clustering of the receptor into clathrin-coated pits, or they can prevent the release of LDL into the endosome as the endosome acidifies. All of these types of mutations in the LDL receptor can lead to familial hypercholesterolemia. And an autosomal dominant form is the one we talked about, where the LDL receptor is secreted. So that's found in about 1 in 500 individuals, that mutation. And about 1 in a million patients will be homozygous for this mutation at birth. If you have familial hypercholesterolemia, your serum cholesterol levels, as you can see, are very high. So serum cholesterol levels should be below 200. That's total serum cholesterol. Also, LDL residence times in these individuals is close to 3 times what it is in a normal individual. And you get cholesterol deposits, and they tend to deposit around tendons, in macrophages, around tendons subject to high shear forces, because those are the places where the macrophages will go to repair the tissue. So you'll get them on the wrist or on the knee, and these are called xanthomas. If you cut into these, they're pure cholesterol crystals. And here's one of them near a knuckle. You get them on elbows. Yeah. In fact, see how red that is? See how red that is? They're just constantly irritated.
Are they removable?
Yeah, they're removable. They scar. Usually it's very hard to remove them without scarring. They also tend to chew up the tendon. They cause a lot of damage. So atherosclerosis results from the formation of these plaques. It's called atherosclerosis because an old term for the plaque is an atheroma. In fact, old books will call plaques atheromas. And this is due to the subintimal deposition of cholesterol in larger vessels, particularly at places where there's turbulence. These can weaken the vessel if it's a big vessel, like the aorta, and it can lead to an aneurysm. Damage to the plaque can cause formation of a clot. That's a thromboembolism. If the clot breaks off, it's an embolus. If the embolus clogs an artery, that's called an embolism. So for example, a pulmonary embolism, if the embolus forms someplace and then it gets stuck in the lungs, that's life-threatening. If it gets stuck in the coronary arteries, it can cause a heart attack, myocardial infarction. If it gets caught in the vessels in the brain, it causes a trans-ischemic attack, otherwise known as a stroke. And if it clogs smaller arteries, it'll cause an infarction, which can lead to tissue ischemia and potentially end organ damage. So the mainstay drugs to lower serum cholesterol levels are statins. Statins were discovered in the 1970s. The first statin was discovered in Japan by Endo Corporation. It was called Compactin. Compactin never made it to market because in animal studies it caused lymphoma in dogs. No other statin does that, but Compactin did. It was just a freak finding, but it was confirmed by other observations. There's no evidence statins cause lymphoma. The available statins cause lymphoma in lab animals or in humans. No evidence at all. You'll still see that on the web. Statins cause lymphoma. Again, the technical term for that is crap. So these drugs inhibit HMG-CoA reductase, and to a lesser degree, HMG-CoA synthase. They have some structural similarities to mevalonate. And because of the way the LDL receptor is regulated, because you decrease endogenous cholesterol synthesis, you upregulate the LDL receptor. So an added bonus of this drug that was not anticipated when they were made is that you increase clearance of LDL with these drugs. That's why they can drop LDL levels so respectively. So here, and I think on your handout I gave you, there's some pictures, some drawings of yeah, there's some pictures of some drawings of statins. Okay, so you can see the mevalonate here. Notice the mevalonate structure here. This structure is found essentially in these. So if you just simply, these R groups, make them hydrogens, you have Compactin. Compactin never came to market. Merck Corporation, under the leadership of Roy Vagelos at the time, was working on lovastatin. Lovastatin was the very first statin ever marketed. It was called Mevacor. It's of course under generic now. It's still used. Then that was followed by the release of simvastatin, which is under generic, but gets called Zocor in its labeled brand. and then pravastatin or Pravachol as the hydroxyl group right here. These are all inhibitors of HMG-CoA reductase. So they decrease endogenous cholesterol synthesis. As you get older, your cholesterol levels tend to creep up sometimes for a variety of reasons. And statins have proven, there's been extensive post-marketing research on statins. It's clear, absolutely clear, statins lower the risk of cardiovascular incidents in people who take them. They do. So here are some other statin structures. The one I want to point out that wasn't on this is atorvastatin here. This is the one that was marketed as Lipitor. On the commercials for Lipitor, you'll hear this statement, Lipitor is the most heavily researched medicine. That's actually correct because there have been so many bloody prescriptions written for Lipitor. There's extensive information on this drug. So this is definitely the most widely, not good. This is definitely the most widely used statin. Now, President Trump, former President Trump, is on rosuvastatin, which is one of the newest statins. This one goes marketed by the name Crestor. This is a so-called superstatin. This superstatin can actually drop your, so let me label it for Amanda here. This one here, this one can actually drop your LDL levels by 50%. That's the reason why Donald Trump, despite his serious love of junk food, has a serum cholesterol level that's still only about 250. And he called his numbers beautiful. I was thinking, you're on rosuvastatin, your total cholesterol is 250 and you're calling your numbers beautiful. Yeah, probably. So, that guy is so full of crap, his eyes are brown sometimes. Anyway, so this is one, and then there's another one that's used now, fluvastatin as well. So this one is not used today in prescription medicine. Neither is this one. This one was pulled because of excessive liver damage. But these are all used: lovastatin, simvastatin, pravastatin, atorvastatin, fluvastatin. These are all used as prescriptions today. So pravastatin is not metabolized by the phase 1 liver enzymes. So simvastatin is a cytochrome P450 3A4 inhibitor. Atorvastatin is not, but it's a 3A4 substrate. So if you're on a drug that's a 3A4 inhibitor, you don't want to be on either one of these. Pravastatin is not metabolized by liver enzymes. It's phase 1 liver enzymes. It's metabolized by phase 2. So you don't have the enzyme drug-drug interactions that you have with these others. So pravastatin is a better drug for certain people. Atorvastatin will drop your LDL up to 40%, well, depending on the dose, because this can be dosed anywhere from 10 to 80 mg per day. At 80 mg, this can drop your LDL up to 60%. It's very efficient. Fluvastatin is a weaker statin. Let's say you're more of a borderline person, then fluvastatin. And then lovastatin is also a weaker statin. And lovastatin is used a lot for menopausal women, because after menopause, some women, their LDL starts to creep up a whole bunch, or just a little bit, whereas as before menopause, their numbers are really good. Then after menopause, and the numbers go up. And lovastatin essentially is good for that because it's a weaker statin and it can take numbers that are outside the range and then put them back in the range. So statins, again, because they increase the transcription of this gene, you increase uptake essentially of VLDL, IDL, LDL. So your LDL transit time goes down, goes way down. Okay, my little story. So I was Mr. Borderline for years. Finally, my doctor said, "You're over 50. Borderline days are over. It's time to put you on." So he put me on low dose atorvastatin. And I had my numbers checked last summer. And so I went from, 202, 201, 204. My cholesterol, my whole cholesterol was 120. So just that 10 mgs in me dropped it like a rock. So even a low dose can work really well in some people. Raises HDL a little bit, not a lot. Now, if you cannot tolerate statins, there's another group of drugs that can be used called fibrates. Fibrates. Okay, now how do fibrates work? Fibrates activate a transcription factor, and to explain this, let me go back to let me go back to my makeshift blackboard. That breeze ruffles my papers. Okay, so in your body, there's a group of transcription factors that are called PPARs. And there's a, can you see that color all right, or is that too faint? Amanda, can you see that? No, she's not in her head, okay. There's a PPAR alpha, PPAR beta, and a PPAR gamma. Now, PPAR stands for peroxisome proliferator activated receptor. So you can see why we abbreviate this PPAR. So PPAR alpha is the target of your fibrates. And what does PPAR alpha do? PPAR alpha regulates lipid and glucose metabolism. It can also help regulate inflammation. And also endothelial function. So this is a very busy transcription factor. Sorry? Alpha, yeah, alpha. Alpha. There. Okay, so fibrates essentially, when they form a complex with this, they activate it. So this plus the fibrate, that activates it. Now, these, when you give them, they decrease the serum triglyceride levels. They also decrease VLDL levels. They usually decrease these by anywhere from 25% to 50%. They also may increase HDL levels. So these are very good for people with very high triglyceride levels. Now on your handout, you have the structure of some fibrates. So here are your different fibrates. Of these fibrates, only gemfibrozil has demonstrated beneficial effects on cardiovascular outcomes. Clofibrate was discontinued in 2002 due to adverse effects. Propofol is not used anymore because of it will lower HDL levels, but gemfibrozil is the fibrate that does have the demonstrated cardiovascular benefit. That's the one that's usually used. Okay, let's go back to the slide. Hanging in there, Amanda? After this, you can take a nap. How's that? All right, so here's the effect of the fibrates. So it increases, one of the genes increased by PPAR alpha is LPL. So you deplete more fats from these particles. You also are going to increase the production of HDL. And then you will also have some increase in LDL receptor production as well. Fibrates on the average are not as potent as statins, but you do get some reduction. All right. So side effects, some people don't stomach them very well. Some people find that their livers start to, LFT stands for liver function test. Liver function test might start to become abnormal. Some people may have muscle issues. They get myositis, sore muscles. The biggie essentially is cholelithiasis. Those are gallstones. So this is a risk with fibrates, not a big risk, but some people have increased risk of gallstones. Niacin is also used. If you use niacin, you have to take really high doses of it. Niacin seems to work on HDL. So you take a gram of niacin, you take it orally. Niacin seems to prevent HDL catabolism, so HDL will last in the circulation for longer, so it's able to withdraw more cholesterol from foam cells, from plaques, from other cells, and what have you. Niacin, when you take it, can cause your skin to flush red because of a histamine release. If you take a baby aspirin half an hour before you take the niacin, you'll prevent that from happening. There are also preparations of niacin that are non-flush niacin because they're gradual release niacin. So Niaspan is the commercial version. Non-prescription forms of niacin have been linked with liver damage. So if you want to take niacin, you should see your doctor and take niacin. Niacin used to be available with statins. That's been withdrawn from the market because no one's been able to demonstrate that adding niacin with the statin increases the clinical benefit. So those particular compounds now, those combos, not available. And then also estrogen. So estrogens increase the synthesis of apolipoprotein A1. So they greatly increase HDL. They also increase the rate slightly of VLDL production. But with estrogen, you also get increased LDL removal. And that's one of the reasons why premenopausal women have a consistently better lipid panel than do men. That's just the way it goes because of estrogen. Okay, these are just some things that can cause low HDL. We talked about Tangier disease. There's LCAT deficiencies. There's ApoA1 deficiencies. Then there's some other kind of unknown things. Oh, it's time, yeah. So, and then just some gene targets. So we finished this, and herein lies the end of the material for next week's exam.
So what where does it, what materials are
If I'm not mistaken, that the fatty acid synthesis stuff was not on the last test. So essentially, Amanda, help us out here. Fatty acid synthesis was not on the last test, was it?
No, it wasn't.
No. So it'll be fatty acid synthesis, lipid synthesis, cholesterol synthesis. So it's a lot of stuff. Yeah. And then after this, we start photosynthesis. Purine biosynthesis. We're getting to that.
also we talked about something, do you think
Wednesday, March 24th? Yeah. Amanda, how does that sound to you?
Yeah,
you
I'm
hear
fine
the
with whatever.
Did you hear the conversation?
Exam on March 24th.
Yeah,
Yeah, that's fine.
that's fine. Yes, that'll work. That'll work. Okay. All right then. Amanda, take your nap. I'll see you Monday. Feeling chipper, I'm sure.